Bless you. I wish you well, and thank you very much indeed.
Thank you. Okay, we now move on. A moment's breath. We now move on to the Emanuel Miller Memorial Lecture, which is an annual lecture delivered. And this year, I'm delighted to introduce the speaker, Professor Dorothy Bishop. It's a privilege to have her here talking today. You've got a biography of her, which I'm not going to reread, but she's Professor of Experimental Psychology at the University of Oxford and has for many years been one of the leading researchers in the field of children's communication, researching diverse areas related to this, from brain function to genetics, to developing some of the tools that we use in everyday practice, certainly I've used in everyday practice. So right across the range. And it's fantastic to have her here to talk to us today about intervention for language impairment. Can neuroscience help? Dorothy.
Well, thank you for that warm introduction. I'm very honoured to be giving this lecture to the association. I was involved with the association for many years as an editor of the Journal of Child Psychology and Psychiatry, and so I have very warm associations myself with the organisation, and I'm delighted to see it sort of focusing its attention on my particular field of interest of speech and language disorders. In preparing for the lecture, I did a little bit of research on Emanuel Miller to think, well, is there any sort of link between what I'm going to talk about and Emanuel Miller, who was one of the founding fathers of the association. In fact, he died in 1970, which was the year when I went up to Oxford to read experimental psychology. So there's really little overlap there. And he was very much somebody interested in working with children, with a particular interest in delinquency, which actually is a topic that some of the talks later today will relate to. But I think if there is a link between my talk and his, it's a strange one, because I'm going to be talking about all this high-tech neuroscience stuff. And I do some of my research in that area. But actually, what I want to use this talk to do is to take a step back and say, what is it actually telling us? And to argue that, yes, it has its place, but that we should put at the forefront of our research endeavours studies of behaviour, particularly when we're interested in looking at intervention, and not be totally seduced by all the neuroscientific things that are going on around us. And I have a sense that this is a message that Emanuel Miller might have agreed with. In terms of what we know about brain scanning, I started my PhD in a department of neurology in 1975 after training as a clinical psychologist at the Maudsley. And at that time, there were children coming through who had various neurological conditions, and our ability to see what was going on in their heads was extraordinarily primitive. But everybody was getting very excited about CT scanning, which was the new thing, because prior to 1972, there was no CT scanning. And if you wanted to know what was going on in the brain, there were very difficult and not very effective and quite invasive methods that you could use. The CT scanner, the history of the CT scanner is wonderful, and I encourage anybody who has spare time to have a look at this article by Filler, because he points out that it was really all a bit of an accident, the development of CT scanning. And it was all down to the Beatles. This may seem rather surprising, but the point is that there's this guy, Godfrey Hounsfield, who was working on quite other things, and he was employed by EMI, who some of you may remember, you're old enough, actually with a company who made all the royalties from the Beatles' great hits. They had so much money from the Beatles that they were able to run a research centre which rather gave people a sort of free rein to try things out. And Hounsfield describes how he went on a ramble one day and thought of the idea of the CT scanner, and they gave him permission to try this out. And he got his ideas to a certain point, and he went to people at Queen Square, neurology department, and he said, you know, we're interested in developing this thing. and he was actually sent away with a flea in his ear for having a completely preposterous and unnecessary idea. And indeed, apparently, the rumour has it that the guy he went to see, who was the head of neuroradiology at the time, was so incensed that he actually phoned up Hounsfield's boss and said, "Don't let that man back in my department ever again." So Hounsfield went off to Atkinson's Morley's Hospital instead, which is subsequently St George's, and developed the CT scanner. And eventually, in 1972, produced images such as the ones you see here, and everybody was amazed and wowed by these, which looked to us like really primitive images of the brain. But of course, this was the beginning of us being able, non-invasively, to start seeing what's going on in the head. And my goodness, we've come a long way since 1972. So that these days, the images you get look more like this. Now, this is absolutely beautiful multicolored pictures showing the structure, the grey matter volume in the brain and how it changes from 5 years of age to adulthood. And one of the key things that studies like this have shown us is that the brain is still developing massively right up through adolescence and into adulthood. One of the other surprising things is that we now know that an awful lot of that change actually involves loss of tissue. Grey matter declines as you get older, so the bluer and more purple your brain is here, it's the less grey matter volume that you have, and you can see that increases with age. So that brain development is very much a process of sculpting the brain and retaining the connections that are functional, but actually getting rid of non-functional connections. And there's a sort of pruning process. So this sort of work, I want to be very clear, I wouldn't denigrate it for a moment. I think it's, you know, we've learned a huge amount, and we're continuing to do so as imaging methods get better and better. And this paper by Guide and Rappaport I particularly recommend as a good review of where we are. But I do think there's also some problems. There's a rather nice little sub-branch of cognitive psychology which looks at how people's willingness to believe things is related to whether you talk about brains. And it's fascinating. In this paper, which has got the wonderful title of The Seductive Allure of Neuroscience Explanations by Weisberg and colleagues, they actually looked at this directly by giving their participants little vignettes, little stories about well-known psychological, well, not so well-known, but psychological phenomena. And they either provided them with a good explanation, or they provided them with a weak explanation that was really rather circular and just a redescription. And this curse of knowledge is a case in point where what I'm showing here is a poor explanation, because they say the curse happens because subjects make more mistakes when they have to judge the knowledge of others. People are much better at judging what they themselves know. That's actually just a redescription of what is meant by the curse of knowledge. So this is a poor explanation. And people recognize it as such, usually, you hope. But what happens if you add a bit of verbiage? Brain scans indicate that this curse happens because of the frontal lobe brain circuitry known to be involved in self-knowledge, blah, blah, blah. Suddenly, what was a bad explanation is now reckoned to be a much more reasonable explanation. If you see these two graphs here, if you've got a good explanation, it remains good, with or without the brain stuff. But the bad explanations are disguised and hidden behind this verbiage about brains. So vacuous and unsatisfactory explanations get accepted. This is lovely literature, there's more of it. Here's another one. The effect of brain images on judgments of scientific reasoning. McCabe and Castell had a similar sort of thing. Instead of giving them psychological phenomenon, they had things that might be like a piece you'd read in the newspaper of describing something. A recent study measured brain activity as children watch TV. It was found that TV watching activated the same region of the temporal lobe as was engaged when they did a maths task. Researchers concluded that children should watch more TV to stimulate their maths skills. Okay, now some people were given that just like this, just as a written statement. Some people were given graphs showing the amount of temporal lobe activation. Some people were given brain scans. And I don't think you need me to tell you which group believed this most. Picture of a brain scan with this text is regarded as much more plausible than the text alone or the text with the graphs. So we see these brains, we see blobs on them, and suddenly our judgment starts to fail us. This is well known by people who sell commercial interventions for children with neurodevelopmental disorders. They put pictures of brains on their website and claim that whatever they're doing, it's affecting the brain, which of course it must be if it is actually doing anything to cognitive skills, because the brain is the organ that does that stuff. But what a lot of these organizations will do is then pull in stuff that they get from some other literature and make it sound as if what they're doing is somehow very specific. This company, Learning RX, I don't know much about them other than that they've got a big presence on the web in the States. I don't think that's so big over here. But this is from their website. They say that they're training the brain, and then they go on that brain training rewires the brain and creates life-changing results. And then they show you these pictures of brains before and after intervention. So this is a normal brain. This is a brain of a dyslexic child before intervention. And notice there's not much in those little circles. And then after intervention, there's more. Now, the interesting thing about this picture is we're going to come back to it later in the talk, because it's not a picture from children who had Learning RX. This is actually from a completely separate study of children given an intervention known as Fast for Word, which I shall have a lot to say about. But essentially, what we have here is a load of guff about brains and rewiring attached to an intervention which has actually not got good empirical evidence, but which is sold to parents to make them feel that, you know, this is something sort of medical neurological. Here's just another example. There's loads of examples all over the web. This one I was asked about recently because they've just opened a centre in Milton Keynes, Sensory Activation Solutions. They have centres all over the world. They offer something similar to what is known as auditory integration training, which some of you may have come across, where you're playing different sounds to the two ears with the idea that you're changing hemispheric lateralization or ability to filter out particular sounds. Again, there's not empirical evidence for this. There's a few studies and they haven't been very impressive, but it's described as improving all sorts of things, attention, dyslexia, speech, social skills, but in just a few weeks. And the point about it is that, you know, the website, again, will have a picture of a brain, green and blue hemispheres, and this is sort of somehow makes it much more compelling than without the picture. So you have to be very, very careful. The commercial people know about our tendency to be seduced, the seductive allure of pictures of brains, and they use it. So my argument here is that anything neuroscience-y just sort of leads to a general loss of critical faculties. And we need to be careful. Now, the point is one can mock, you know, some of these advertisements and say, good heavens, you know, nothing that they say here is right. But I want to argue that this loss of critical faculties doesn't just affect the lay people. It affects scientists and practitioners as well, and those of us, in a sense, who should know better. And the standards for acceptance of research that involves some sort of neuroscience are actually rather low compared to standards in other fields. I'm going to focus on one paper, and you might think this is really rather harsh to single out one paper, and it is. I mean, it's because it's got a lot of problems, and I'm going to go into them, and I don't particularly like to do this, but this paper was published in a top journal, the Proceedings of the National Academy of Sciences. Most people would kill to get a paper in the PNAS, as it's known, because it's regarded as a real mark of distinction. The authors are very eminent, and this paper's been cited 270 times, which is a lot, so it's been influential. And I thought, well, maybe people are citing it to say what's wrong with it. But no, I mean, I was able to look at about random 50 of these citations, and there was one of them that picked up that there was a problem with the methods. So in general, this has been immensely influential. The claims are that it's a study of looking at how this intervention, Fast for Word, which is probably known to many of you, affects the brain. So, and there were 20 children with dyslexia. They were given functional brain imaging before and after this remediation program. And the idea was that on the one hand, you could show that the behavior of these children in terms of performance on language and phonology and reading tests improved after the intervention. And there are also claims about the brain, that the children with dyslexia showed increased activity after the intervention in many brain areas, and that there's a particular region, the left temporoparietal cortex and left inferior frontal gyrus, which actually became closer to normal children. And here's that picture again, only this time with some colour. So this is the study that the picture comes from, where you see over here, in these circles, in the more frontal and posterior regions, you see nice red, which indicates activation increasing. Here you see children with dyslexia before remediation, where clearly not a lot is going on, and then after remediation, wahey, the brain is now pink in those regions, much more like the typically reading children. So you can see why people got excited about this. They also claimed, to really clinch it, that this increased activity in this region, in the blue region, the posterior region, was correlated with the improvement that you would see in language in the children. And here's the scatter plot that shows that improvement. But there were a number of things that got forgotten in this study. And again, I sort of think, well, it's because, you know, you've got these brains. We all sort of think, oh, look, the brain is changing. But in fact, you've got to look at this rather more carefully. First thing is they didn't have a control group. So they just had children who were tested before and after, dyslexic children who were tested before and after this language intervention. They had some typically developing children who were not intervened with, who were seen on 2 occasions, but they didn't have what they should have had, which is a control group that was similar to the dyslexic children who were treated. Why do we need this? Now, there's a sense in which this is sort of intervention studies 101, but a bit like John Burko, I sort of have come to the conclusion if you've got something you need to say, you say it several times. And I've been banging on about this for quite a while. But what astonishes me is how many people just don't seem to believe it. They sort of may understand intellectually what I'm saying, but they just don't really believe that you could see changes after intervention that weren't due to the intervention. So I'm going to just hammer on about how this can happen. An obvious reason why you might change after an intervention is because you've got older. So that if I measure your foot size before you do fast forward and then again after you've done it, it will be bigger. It's not anything to do with the intervention that you've had in the interim. The plausibility of that as an explanation varies according to what measures you're using, but for many measures that are used, this is plausible. And in the brain as well. Now, the other thing that is very clear from the way people talk about these brain results is that there seems to be an implicit assumption that the brain, because it's a physical organ, is somehow not going to change unless you give it some intervention, and that you know, it's sort of there as a static thing. This is completely untrue. You can see changes in the brain with maturation, as evidenced by this series of images from Jay Geed's lab. So we can't just assume that any change we see is related to intervention in either behavior or in the brain. There's also practice effects, and my favorite story about this is my experience with the Wii Fit, a sort of sad attempt to try and improve my fitness levels. I've got one of these things where you stand on this balance board and the computer says to you, well, you know, now stand on one leg and how many seconds without wobbling and so on. And then the first time you do it, you come out and you've got a brain age of 73. It's pretty depressing. You go away, you come back the next day and do it again without having done anything healthy in between. And your brain age has improved by 30 years. This is purely because you've got better at doing the test. It's nothing to do with a genuine improvement in your abilities. And people tend to assume that this only applies to things like Wii Fit, and they tend to forget about the fact that it can apply also to things like language tests and things like that. And I'll show you some evidence for this if you don't believe me in a minute. It also applies to some extent to the brain. And often we don't know how important this is because brain imaging is so new. But for example, there's a whole branch of research on brain imaging that's interested in how the brain responds to novelty. Well, clearly, if you get brain responses to novelty, that means that if you do something twice, the first time round, you'll get a different pattern of brain response to the second time round when it's no longer novel. So again, we've got to get away from the idea that the brain, you know, there's a very reliable and consistent signal coming from the brain in imaging studies. The third reason why your score may improve is the most technical one, but it's also the most pervasive, which is to do with what happens if you select your participants on the basis of a low score on a test and then assess them later on to see what they're like on that same test. You then suffer from a statistical artifact called regression to the mean. which is described by Campbell and Kenny as inevitable as death and taxes. There's a nice paper by Zhang and Tomlin describing this in how this can affect results from longitudinal research designs in the field of speech and language. And how it works is like this. This is my best, it's very hard to explain, and I've tried this for many years, but I think this is the best I can do. So the idea is that this is simulated data from, these points are individual people that I have just simulated by simulating a normal distribution. Scores at time 1, mean of 0, so these are like Z-scores for those of you into those things. Let us suppose that our measure is really unreliable, so it might be standing on one leg, but it may be influenced by all sorts of things like whether the cat's run into the room or something. So you get a distribution at time 1, you get a distribution of scores at time 2, which is very similar, but the relationship between the two isn't very good. Okay. So that's showing you some people go up, some people go down, etc., etc. The average score at time 2 is exactly the same as the average score at time 1 in this simulation. So there isn't any practice. But now look what happens if you select the people who did badly on time 1 on this measure. There they are, just a few of them. What's their average at time 1? Well, it's low because you've selected them to be poor, but by time 2, their average is much better, just because this isn't a reliable test, and the anticipated score for everybody is going to be the population mean. There is a distribution of scores at time 2, just as there is at time 1, but if you just take people selected to be bad at time 1, the odds are that at time 2 they'll do better, because the test isn't perfectly reliable. Now, if you get a much more reliable test, things look much better. So again, we've got our group of people at time 1, group of people at time 2, and you can see that this is a reliable test and that the lines for each person tend to be connected in a more coherent way. This is a simulation where there's a 0.8 correlation, but you still get regression to the mean. So these are people who are bad at time 1, mean score, at time 2, mean score. It's not a big difference, but it gets better. And it's completely predictable from two things: how extreme the people are when you select them and how good the reliability is of the test. But essentially, you get regression to the mean. And if we think about our psychological measures, things like language tests, where we know what the test-retest reliability is, it's often not more than about 0.7. 0.8 is doing quite well for most psychological tests, and sometimes it's lower than that. So you're going to see improvements that are totally artifactual and to do with the way you select your participants and follow them up. Okay, last one. This could go on all afternoon, but placebo effects. If you ask people why do we need a control group, this is the thing that most people will say, yes, you've got to worry about placebo effects. As you see, I would say that these other things are actually probably more likely to be important in the sorts of interventions we're talking about. But certainly placebo effects are a source for concern, whereby a child may start showing improvements simply because they're getting the extra attention and everybody's pleased and the parents are supportive, without the intervention really being responsible. It's more the sort of general ambiance. So this is why, I've banged on about this now for some minutes, but this is why you really do need a control group, because a control group can allow you to consider the effect of all those things. You may often not know which of those things it is you're controlling for, but you have a control group who are also tested before and after, but who were selected, importantly, the same way as your intervention group. That will give you an idea of just how much change can be due to these spurious factors. A lot of people are very unhappy about the idea of an untreated control group. We can argue about that if you like, but more commonly in mainstream medicine it's become usual, in fact, not to have an untreated control group, but to have an alternative treatment group, but where you can set this up ideally so the alternative treatment isn't intended to perhaps focus on the specific thing that your treatment is working on, but you're not denying children an advantageous therapy. But I would argue that it's crucial to have a control group if you want to know whether the improvements that you see are meaningful and not due to these like the 4 horsemen of the apocalypse that come in and mess up any sort of study that you otherwise want to do. Just in case you don't believe me about this, here's some hard data from my own studies. I did a very small-scale intervention study published in 2006 trying to train language comprehension with no success on a computerized intervention. Now, what is interesting is you may say, well, what is not no success? Group S and Group M both had different sorts of treatment here. And as you can see, their scores went up. This is a scaled score on TROG, where the expected mean is 100, but they're gaining about 10 points. It's only when you look at the untreated control group that you realize everybody goes up. It's not just the treated group. So without that untreated control group, it would be very, very easy to conclude that the treatment had driven this effect. But you need the control group to really be confident as to what's going on here. There's another thing that is often overlooked in studies of this kind, which is that even when you've done everything properly and you've got your randomized control group and your intervention group, and you compare them at outcome, you can have another reason why you may get a spurious result, and that is to do with selective dropout. People who drop out of treatment are not very often a random subset. There's likely to be two reasons why, well, three. They might just move away or something, but otherwise they may be people who are dropping out because they don't think the treatment's working, and they're sort of fed up with going on and on with it when they find it difficult and they don't get on with it. Alternatively, they might be people who are actually doing so well they don't think they need the treatment anymore. But they're not likely to be random. And again, an illustration from that study I just showed you, we only had 3 dropouts in this study, but look at their mean TROG score at pre-test. This is a clear case where the kids that dropped out were the kids that were doing very, very badly to start with. This was an intervention where they had to respond to spoken sentences by moving pictures around the screen. If their comprehension was that low to start with, they were going to find this hard, and they dropped out. Nevertheless, at post-test, look at how well they did. Up they go. Again, this is just the general effect, I think, of practice, regression to the mean, etc., etc. So we really do need to worry about methods in this area, and I think not enough attention is often given to it. And for the Temple et al. Study, they didn't have a control group of dyslexic children randomly allocated to their treatment and control. What they did have was a group of children who were typically developing, and they were also tested twice over the same interval. But this is really problematic because it's not an adequately matched control group, and the changes in behavior in the treated children could be due to all the factors I've just been talking about, and some of the changes in the brains could also be due to some of those factors. So it's really problematic that they are trying to rely just on a pre-post-test comparison. Just to bang it home even more, here are some other data on how language test scores can change even without intervention. A study on the KELF screener back from 1996, it's an early version, just didn't take children with problems, just took a big group of children from a preschool over a 7-month interval, tested them once, tested them again. They had a massive improvement in scores on the second test, an effect size of nearly 1 standard deviation. Gillam did a randomized controlled trial of Fast Forward, which included 3 treatment groups, which are shown here in blue, maroon, and yellow. Well, blue, maroon, and yellow, yeah. And a control group who also treated, they had an intervention, but it wasn't designed to improve their language skills, which are shown in green. Again, if you ignore the green group, then everybody's doing well. This is the improvement in standard scores after treatment at post-test, so there's a big leap in standard scores there, and then improving more and more as time goes on. But note that the children who were in the control group are doing the same thing. So you need that control group to be able to conclude that, to be able to interpret the improvements that you see in the other groups. Strong and colleagues published in the Journal of Child Psychology and Psychiatry last year a meta-analysis of fast-forward studies that had properly controlled designs, and there are about 5 or 6 of them, can't remember exactly, but just found that on average on this meta-analysis of well-designed studies there was no benefit of fast-forward relative to controls. So meanwhile we have the Temple study, which is saying, well look, we saw the KELF-3 score go up and their effect size was about 0.6. And they say, well, there we are, we've shown an improvement associated with fast forward. But as you'll gather from what I'm saying here, that sort of effect size is not uncommon in children who are just tested twice without any intervention in the interval. It's depressing, but it's something we need to take into account. There's another problem with this study. When you do brain scanning, or indeed electrophysiology, like event-related potential studies, you have a lot of data. But brain scanning, you have a hell of a lot of data. You have tons, people are delighted at all the data until they realize, my God, what am I going to do with all these numbers? So you've got, the brain is divided up into little cubes called voxels, and from one brain scan, you can have up to 130,000 of these. So you've got a problem of deciding how to interpret any differences that you find, because if you do enough comparisons, as any statistician will tell you, something will pop up by chance. And Poldrack and Mumford in 2009 got very exercised about this and wrote here, and indeed elsewhere, about how important it is to employ accurate corrections for multiple tests. You've got a large number of voxels, you'll get results significant by chance. And this problem is well known, but unfortunately, many journals still allow publication of results based on uncorrected whole brain statistics, which is what the Temple et al. Study did. And if you look at their table of results and you show it to somebody who really knows about brain scanning, they will say that this has got a set of results that has a high probability of being chance. The most convincing study, again, for those of you who really just find these things very hard to believe and don't like statistics, is the study of the dead fish. This dead fish, it was an Atlantic salmon, and it was used to test a brain scanner. These people had just taken delivery of a brain scanner, and they wanted to try it out. So they stuck the dead fish in it, and just for fun, they ran their research fMRI paradigm with the dead fish. And so the dead fish was presented with trials where it was supposed to judge whether faces were happy or sad or something like that. And at the end of the day, when they ran uncorrected whole brain statistics, they got areas of the dead fish's brain that lit up. And they pointed out that this could not be meaningful. And so this is a very nice study that I think hammers the point home more than any amount of statistical reasoning. Okay, there's another problem, which has recently been written about in some of the neuroscience literature, which is that a lot of neuroscience studies, if they're trying to compare two groups of people, they will say, well, group A, we tested them on this condition, and look, their brain changed, this region lit up and that one didn't. Group B, it was the other way around, and therefore the two groups are different, which sounds all right, except that it's sort of, again, it ignores basic statistical reasoning, which is that you have to take into account how much variance there is in the groups, and you really need to look at, if you're trying to argue, for example, that children, two groups of children show different profiles before and after an intervention, you really need to show that there's a significant statistical interaction which takes into account not just the mean changes, but the variability. The Temple study, as I said, didn't have a proper control group, but they did try and argue that there was a sort of interaction in the sense that the dyslexic children's brains became more like the typical children's brains at time 2. But they didn't analyze the interaction. What they did do was to show you that picture. And that picture is so compelling that you just show it to somebody and everybody goes, woo, the brain looks more like the controlled children. But what those blobs don't tell you is really how much variation there is among them. So children vary in terms of how their brains are activated. And this is not necessarily a meaningful result. You need to do the proper statistics before you can be confident about it. Fourth problem. Seemed very compelling. You can say, well, this is all very well, but they did actually show that the increase in left temporoparietal activation correlated with improvement in language in these treated children. What better evidence than that could you have? Well, there's a problem. First of all, they did lots of correlations, and it's the same problem that we've already thought about, where you do enough statistical significance tests, something will pop up. They had a lot of different language and literacy tests, and they had an awful lot of different brain regions, and they present this as the result that was most interesting. But if you look at it in the context of how many comparisons they did, it's not terribly meaningful. But even more worrying, when you actually look at the scatter plot, you think something odd's going on here because of those two guys. If you take those two outliers out, the correlation falls to almost insignificantly different from 0, very low. And the point about those two guys is they're not just outliers, they're people who are actually failing to improve in language because they're getting a reduction in this temporoparietal activation. So it's not the case that the more activated you get, the more your language improves. It's rather that there's a couple of people where something odd has happened and the activation has gone down. So this is the sort of thing, again, that is really quite worrying and would make most people query whether this is a meaningful result. Now, I am not a brain imager. I do some electrophysiological studies, and I work with people who do brain imaging, but I sort of was reading all this and with quite attention to detail, and I was getting more and more fascinated by all the problems with this paper, and I thought, well, I'll write a little blog post about it and see how the world reacts. And it's been really interesting. So I posted this blog earlier this week, and I nearly fell off my chair because I got this response on Twitter. DVB, that's me, tears apart a 2003 PNAS study that I'm a co-author on. And she's spot on. Yes! This is Russ Poldrack, who's like, you know, a very eminent and well-respected person who really does know about imaging. So it would seem that at least one of the authors of this paper actually agrees with me that there are problems with that study. Nevertheless, it's in the literature, it's being used to market fast forward, and people see these pictures of these brains and think, yes, this works, it changes the brain. So we do have a problem, and I'm glad that actually what's happening now is there's more debate about these issues, and I have to say I'm incredibly impressed that Poldrack is so frank and honest about it all and keen to engage in debate. But it is an issue that one says, well, why did that study get accepted when it had so many problems in the first place? Some things have changed with the passage of time, but things like not having a control group are fairly fundamental for a top journal to accept a paper in that situation is rather odd. It's not just that paper either. I mean, I focused on that one because it is so influential. But here are 5 other studies that I found. There's quite a few studies looking at training and brain impact of dyslexia training. These are all ones that were specifically interested in oral language, children with oral language problems. As the Temple paper, although it talked about dyslexics, it was using an intervention for oral language problems. And the bottom line really is that, you know, the Temple study is by no means unusual in terms of the methodological problems that it has. These studies vary in the interventions that they looked at. Some of you may be familiar with aerobics, which is another computerized intervention. The PICO study was Finnish and used a Finnish group, phonological discrimination. A couple more studies using fast forward, some using a narrative intervention, and brain measures including ERP, MEG, magnetoencephalography, as well as functional imaging. In fact, the functional imaging study listed here is the same group of children seen on the same occasion as the Temple study, but undergoing a different fMRI procedure. I checked with the lead author or the senior author because they didn't actually tell you that in the paper, but it just looked as if it was, and it is. So, but all these studies have, I won't bore you by sort of going through them in excruciating detail, but they all have limitations. So I haven't found a single study that is free of these limitations, and most of them have several limitations. They have very small samples quite often, less than 10 children per group. 5 out of 6 studies lack proper controls. 4 of them, clearly you could argue there's this data dredging element, which is this business of just gathering tons of data and then sort of you've got the opportunity to find something in there. Inappropriate statistics were very common, and we're not talking about sort of really fancy, difficult statistics here. We're talking about fundamental things like testing for an interaction effect, which for most researchers ought to be sort of pretty basic. The other thing I found interesting is that people are launching into these intervention studies without first demonstrating, just in a simple behavioral study, that the intervention is effective, which seems to me the wrong way around, because these brain studies are difficult to do with kids. They take a long time. The kids often don't particularly like being stuck into scanners and having electrodes stuck on their heads. And particularly with scanning, it's very expensive. So people are doing this without first checking that the intervention is effective, which seems very odd, because why do you bother to do all that if you really don't yet know that your methods work? And I think often people just, again, believe that the methods work on the basis of pre-post-test comparisons without really appreciating that without proper controls, this isn't going to be very impressive. But suppose, this is a thought exercise, let's suppose that somebody had done a study that even I was happy with. This horrible woman who's such a killjoy comes in and sort of has a go at people. What would a well-conducted study with decent number of subjects and proper controls and proper advanced a priori hypothesis, what would it tell us about intervention? Because this is the reason for doing these studies, that they're going to show us how intervention changes the brain. Well, I wonder if it would tell us what we think. I think the reason people are doing these studies is because people think like this. This cartoon captures it exactly. You know, you see a brain scan and you think it somehow told you something more fundamental than if you actually have a behavioural measure. So for those of you who can't see this, it's your car keys are under the sofa and you like to wear women's shoes on the basis of what you see on a brain scan. Newspapers continually suffer from this error which Poldrack has called reverse inference, that if you see something on a brain scan, it somehow tells you something about the behaviour. Here's some headlines. I had a little go about this on my blog. I actually just put into, it's a useful exercise to do. You type into Google the phrase, brain scans show. And this is the sort of thing you come up with. Brain scans show children with ADHD have a faulty off switch for mind wandering. Do they really? I mean, you know, would I recognize an off switch for mind wandering in the brain of a child? I mean, come on. Future angst. Brain scan shows uncertainty fuels anxiety. I think if you want to know if uncertainty fuels anxiety, you don't need the brain scan. Rejection really hurts, brain scans show. Brain scans show how multitasking is harder for seniors. For all of these, you could just remove brain scan show. The brain scan doesn't show it. The brain scan might show you a correlate of this, but the notion that the brain scan somehow is getting at the behavior. The way to, Max Coltart has been very vocal on this and pointed out that, you know, suppose you had a study where you did a brain scan and an intervention study, and what you did then was to, what you actually found was that the intervention improved a child's language, and you could see nothing on the brain scan. Would you decide that the intervention wasn't any good? Of course you wouldn't. What about if you did got the opposite result? If you found that the intervention produced changes on the brain scan, but actually the language didn't change at all? You wouldn't think the intervention was wonderful. I mean, it's a sort of weird thing that people are sort of so fixated on these brain scans and what they can show. So I think there's a real problem in this. This is this seductive allure which affects the press massively, but it's also affecting how we think about things. So my main message today is really that we need more behavioural studies if we really want to understand intervention and we really want to get better interventions. What we need are more studies of the processes that affect children's language learning if they have language problems. And these will be behavioural studies, not brain studies, that are really going to be important. I'm going to briefly describe some work that we're doing in Oxford, which my postdoc Julie Hsu is lead author on. We've only just finished gathering the data, and we're hoping we'll write this up later in the year. And I'm not really presenting this as a major presentation of this study, but just to give you an idea of the sort of thing that I think could be potentially helpful in informing interventions. So what we're doing is teaching children new vocabulary, and we're teaching them real words because we think these kids have got enough problems as it is without us teaching them artificial vocabularies and things. So we have to find words that are really unusual. So these are animals, and even I can't remember half the names, and they're very weird things. There's a dugong, which I do recognise, and I think that's a tiwi or something, and that's a genet, and that's an ii. Anyway, they're things that nobody knows when you start. You show them the pictures with the names, and it's just a sort of paired associate training, which is dressed up in a computerized game. They hear a word, they get an array of pictures. If they pick the correct one, they get reinforced. And fortunately, we've been funded by the Wellcome Trust to do this study, which is really very hard to do because you have to keep going back day after day, because we want to look not just at can they do this, but at the process of learning. And this is what we've found so far. We've got 3 groups. What you see in red here is a group with SLI. What you see in green is chronologically aged matched controls of similar IQ. And the language LA group is a language age matched group who are younger children who starting out have comprehension levels that are similar to the SLI children. And this is a result that really quite surprised me because what I had thought would be the problem would be that learning of vocabulary would be slower, and so that the slope of that line would be reduced in children with SLI. Whereas what you can see is that everybody's really learning at pretty much a similar rate on this task, but the starting point is lower and is equivalent to that of much younger children. So right from sort of day one, these kids don't pick up so much. And that suggests that maybe for this sort of vocabulary problem, we need to be focusing more on initial aspects of learning rather than worrying too much about what happens later on. If we could bump up this sort of intercept term here on this, they would look much more like normal children. But it's not that they're forgetting what they've learned, it's just that they start at a lower baseline. But then there's another task which we used, which I wanted to look at because I'd looked at something similar in this intervention study that I had previously mentioned before that didn't seem to succeed in training children to improve their comprehension of grammatically complex sentences. The sort of sentence that we would give children would be things like the apple is before the horse, and we were using above and below and before and after and so on. But little prepositions that just depended on word order for their accurate interpretation. So the child would see this picture on the screen and a number of items that they could then click on and get them in the right order, and they can get feedback if they get it wrong, and Bart Simpson is just there to provide a bit of reinforcement. But what was interesting is that the result looks very like what we saw before and rather different from vocabulary learning. This seems like a trivially easy task, and you would think if you bang away doing this day in, day out, surely sooner or later you would grasp the meanings of these prepositions and how the word order is critical in determining the meaning. But what we found was that there was this sort of flat lining. So the red is the SLI here. I've lost my little arrow for a bit. Here it is. But here are the children with SLI. And what you see is that for typically developing children, this is ceiling effect. The task is trivially easy, which is what we had suspected. For language age match controls, it's also pretty easy, and they actually do get from being very good to being very, very good over the course of the training. So they're more like we would anticipate. But we've got this sort of failure to improve. And the language-impaired children are not just guessing. They know what these terms mean. They're above chance level, but they don't seem to automatize their knowledge of these meanings and just sort of be able to rapidly, after all these days of training, just get this workout which is before and after, which is above and below or whatever. So this to me is really quite interesting. What is limiting this aspect of learning, which seems really quite important for language acquisition? And if we can do more studies to actually see under what conditions can we improve this learning or what is hindering it, this could be useful in I think, developing interventions for the future. What about the brain, though? Shall we come back to the brain for a bit? Because I'm talking about, well, I think these studies just looking at correlates of improvement are not necessarily particularly helpful. Of course, what people are also talking about, and is much more potentially applicable, is trying to do things to the brain that directly affect the brain. So instead of just measuring what happens when you do a behavior intervention, can you do something to put the brain in a better state for learning? Now, this is real sci-fi stuff, but I want to just mention three things that I think over the next 10, 20 years may start to become more prevalent, but which are all very uncertain and all very controversial also, because once you start talking about changing brains, people start to, rightly sort of worry about what you're doing if you're doing this with children. But let me just say about the things that I think may, in the future, come to pass. Smart drugs have been around for a while, and we're all used to the idea that many children with ADHD are given medication in the form of Ritalin. But what's happening these days is that other medications are coming online which are seen to have fewer side effects, and where claims are being made for things that might specifically enhance memory and attention. And what's interesting is the extent to which these are now being used by students at our top universities to help get them through their revision and their exams. So Barbara Sahakian has talked about a study that was done at the University, I think, of Cambridge, which just asked people, do do you know what these drugs are? Have you ever taken them? And it was quite astonishing. About 20% of students were taking them to improve their performance. Some of them are taking Ritalin, but modafinil apparently is the one of choice. It has very few side effects and seems to make you better able to concentrate. Now, as far as I know, this isn't tested for children's learning difficulties, although there are other drugs in the past that have been have been applied to children with dyslexia, but not received approval for being used. But it's an interesting idea that if a child has got a problem with learning, maybe some drug that actually alters their ability to retain information might be something you could use. But it's also something that people find deeply concerning. People are worried about giving drugs to children. A lot of people don't like drugs for ADHD, and there's ethical concerns too about not knowing what this holds in the longer term. But I suspect that this will definitely start being around, and you may get people asking you about whether their child should have these or not. And I think we just need a lot more research on whether this is a good thing. Rather more benign and less controversial is the idea that you can also use a sort of biofeedback known as neurofeedback to alter brain function. And again, where this is most applied has been with ADHD. So increasingly, people have realized that if you look at the electroencephalogram, you can see oscillations at different frequencies of electrical activity in the brain, and that some of these frequencies are associated with particular mental states. So alpha is associated with being more alert or not, and there are different sort of frequencies that you can try and maximize. So people are developing games whereby you get, you're perhaps controlling an animal on a screen by just sort of trying to think in a way that makes it move forward. And what's actually happening is you may be maximizing a particular frequency of oscillation. There's some promising trials of this for ADHD, but it's still in a very early stage, and we've got to be very careful that there can be people who are going to try and market this before we're really certain whether it works or not. And as far as I know, it hasn't yet been tried with other neurodevelopmental disorders. But again, I see this as something that's coming, and I sort of have an area of sort of skeptical interest that maybe this is an approach that could possibly have some promise, but we don't know. And the last one, electrical brain stimulation. Now, when the newspapers talk about this, they often talk about giving people electric shocks, and they liken it to ECT. But this is very, very different. This is having a couple of electrodes, which are shown here on this head model, one at the front, one at the back, but you would put them on different places depending on what you were hoping to achieve. And this is just a unit that is equivalent to a 9-volt battery. So it develops a very low current. It creates a very low current between the electrodes. You don't get a shock. You may not feel anything, or you may feel a slight tingling when it's switched on. I've had this done to me, and with electrodes over the occipital cortex, the visual cortex, and you see phosphenes, you see little sort of flashes of light. I'm still standing. And this, a lot of people didn't believe that this would do anything because it's such a small current, but there have been animal studies looking at what it does neurologically, and it does seem to affect the membrane potential so that neuronal excitability is altered. And there is increasing interest in the idea that this can enhance learning. It doesn't have a long-term effect, at least it hasn't been done in conditions where it will. It has an effect at the time you're stimulating. So what people are interested in is trying to train people to do things while the stimulation is on. And this is being increasingly researched as a possible adjunct to treatment of motor problems in patients after stroke and acquired aphasia. It's also some applications that it might improve language. Nothing, as far as I know, is known about its effects on children. I know there is one group in America that's using this method with autistic children, but it doesn't look as if it's sort of in a research-based way, and it's a little concerning that it doesn't seem to be very heavily regulated. But again, I mention these as just things that I think in 10 years' time we may see more of. You need to be aware that they're going on. I think they're all methods that are not wacky in the sense that they all have potential, but they are also all methods that do raise ethical concerns and where the evidence base as yet is very, very early days. So just to conclude though, what do I hope for the future? I've sort of argued that our obsession with sort of looking at brain correlates of intervention, I think, is overblown. I don't want to badmouth all brain studies of children with neurodevelopmental disorders, but I do think if you think about the sums of money that are spent and the sort of studies that we actually need, these are perhaps not the studies that are most welcome at this time. I would say if we want to work on interventions, we need a lot more work to really look at the basis of the problems that we're trying to intervene with and see how we might be able to alter children's language. And we need then proper randomized controlled trials to develop efficacy to demonstrate efficacy of interventions at the behavioral level rather than sort of rushing into the imaging. And I know that later today we're going to have some more other talks that will actually illustrate this, which is great. But that's my hope for the future, and I hope Emanuel Miller would agree with me. We're here live. And I just end by saying thank you very much for this opportunity to talk to you.
We've got time for a couple of minutes of questions before coffee.
Yeah.
Professor Tommy Mackay, I'll shout so everyone can hear. I enjoyed that very much. I think an additional very good example of how brain scanning can be used to give the reporting of false results is the way the press picked up on the brain scan studies in the diagnosis of autism by Dr. Christine Ecker and colleagues at Case College London recently. And the BBC News and other press picked up selectively one small part of the statistics and said that this new brain scan was 90%
Yep.
accurate in diagnosing autism, whereas if you analyse the whole statistics, it's somewhere between 2% and 4% accurate. Just an additional example.
Yes, I followed that. I'm very interested in this general area, as you'll gather, and I did follow that story and did indeed blog about it. So there is a Bishop blog coming to the conclusion, as you have come, that the papers, if you do anything about the brain, the newspapers tend to immediately want to translate it into a story. Either this will help diagnosis or this will help treatment. And when you think about the logistics of it, are we really going to be shoving children into brain scanners in order to diagnose autism? It's rather easier to do it on the behavioural level, not to, let alone more accurate. So I think it's sort of, sometimes it's hyped by the media, but it's also, I don't know, the researchers themselves sometimes seem to not really quite think it through as to what the implications of their work really is and hype it up a bit.
One last question.
James, I've got one, Dorothy, which is actually relates to your fast-forward comments. I'm not so interested in the poor studies, which people make outrageous claims from, but there have been a number of studies that you suggested, systematic reviews, where a systematic review picks them up, where good quality studies don't get positive results.
Yes.
And given your comments about neurofeedback,
Yes.
and given those studies were about biofeedback, I wonder whether you would speculate about the neuropsychological reasons for them not getting results.
Sorry, for who not getting results?
Those trials not getting positive results.
Which trials?
The trials of Fast Forward.
Fast Forward?
Yeah, specifically Fast Forward. I just wonder whether you've got any thoughts about why they didn't get results.
Well, it's usually the case that, there's a horrible cycle that new interventions go through where they come on the scene and are greeted with great joy. You know this better than me. And then, everybody gets excited and they start to sort of doing it. And then people come along and say, right, we're going to do some proper trials. And at that point, the effect evaporates. So I think it's purely that these are, it is to me rather remarkable, given how much Fast Forward really sort of hammers children day in, day out. This is 90 minutes a day, 5 days a week for 6 weeks. You would think it would do something, and I'm quite surprised it doesn't do more than it does. But I think, it clearly isn't effective on the sorts of outcome measures that they're talking about. And I think it's simply that people are impressed by seeing changes in the children, and they attribute the changes to the intervention, which are probably not due to the intervention. The one caveat there is that, and this again is something that happens with all sorts of treatments, is that there's a complete mismatch too between the objective data on children's language outcomes and the extent to which parents and people, sometimes the children themselves, like it. Typically, these are if your outcome measure is, are parents satisfied with the intervention? The answer is yes. They love it. That's been shown for a number of different interventions, which again, you look at the objective data, actually your child hasn't improved any more than you would expect from a control group. And again, I think there's this tendency to see change and to attribute the change to the intervention, and because what you never see is what's going on in the control group who didn't get the intervention.
I'm afraid we're going to have to stop there. Professor Bishop will be around at the coffee break.
Thank you very much.